Alright, this is the DNA replication video where DNA will be replicated. So we're going to start out with the nitrogenous bases, which is thymine, adenine, guanine, and cytosine, which code for amino acids in translation and transcription and translation. And three pairings called anticodons. And then you have your 3', 5', 5', and 3' on different ends because DNA strands are anti-parallel to each other, which means they're parallel but in opposite ways. And then adenine, thymine have 2 hydrogen bonds, as you can see right there and right there. And then cytosine and guanine have 3 hydrogen bonds. And then thymine and cytosine are pyrimidines, adenine and guanine are purines, because that's 2 ring to 1 ring. And then here's the sugar phosphate backbone for DNA. And then after that we have origins of replication, which actually start DNA replication, because those open up the replication bubbles, and the replication bubbles form because of helicases unwinding DNA, which then puts stress on the farther ends of the DNA that hasn't been unwound yet, which is where topiceromes come into play by taking off the stress from the unwound DNA, because it would also want to unwind, but it's not ready for that. And then also, after the helicases break the hydrogen bonds of the nucleotides, single-stranded bonding proteins bind to the nucleotides to make sure that the DNA doesn't repair to each other and prevent the replication of the DNA. Then after that, you have RNA primers and DNA polymerase 3s on the leading strand that code from the 3' to the 5' end, making the next nucleoside triphosphate, which will be the next part of DNA. That will make the next DNA molecule. Then after RNA primers connect to DNA polymerases 3, and then it allows the DNA polymerase 3 to replicate the DNA or produce the other nucleotide pairing that it needs. And then DNA polymerase 1 acts on the lagging strand, and what it does is it breaks off the 3' and 5' to get an OH group on there, so that the DNA polymerase 3 can replicate it, because it only works from 3' to 5' because of that OH group. And then what's produced from the lagging strand is called the Okazaki fragments, which is pieces of RNA that need to be put together by ligases, which will produce the bonds for the sugar and phosphate backbones, which will put together the RNA, which will then bond with the other piece of DNA to make a full functioning DNA molecule.